Thank you for that very gracious introduction, Dr. Brinkley. It's truly an honor and a privilege to be here and be part of this very august body for a wonderful two-day symposium. I don't know that any other state could pull together this array of activities under one particular subject type like Texas does, so my hat's off to you. I'm quite jealous. So in the next hour, you will hear from myself, from Dr. Mitchell Lazar, who is professor of medicine and director of the Institute of Diabetes, Obesity, and Metabolism at the University of Pennsylvania, and then from Dr. Mauro Ferrari, who is the CEO and president of the Methodist Hospital Research Institute. And what we'll be talking about today is under the rubric of energy, medicine, and nanotechnology. And we really have three very different subjects that the three of us work on, but we tried to pull them together in a common theme that I would describe as discussing an increasing number of human diseases that are being linked to the regulation of cellular energetics and metabolism. And this spans the spectrum of human disorders, from neurologic diseases to hormonal disorders, cancer, musculoskeletal disease, and cardiovascular disease. And this is just a partial list. And what we'll talk about today are various insights into disease mechanisms that have come from the study of cellular metabolism and energetics, and how this has led to opportunities for novel therapeutic approaches. What I will focus on are some recent insights into cancer and metabolism. And many of you know this is a topic that has become quite popular in the cancer biology field, and there's a lot of things that we could talk about. I'm actually going to very much focus my comments on some recent data that provides unexpected links between classic DNA damage signaling and metabolism, and through its effects on mitochondrial function and how that impacts cancer development. Dr. Lazar will talk about hormonal signaling metabolism, with a particular focus on using thyroid hormone as an example of the impact of hormone signaling on metabolic physiology and its disturbance leading to pathology. And then Dr. Ferrari is a world-known expert in nanotechnology, and he's going to talk about how cellular organelles, including the energy center mitochondria, can serve as models of nanoparticle design, ways to deliver macromolecules to cells, and how nanoparticles can be used to selectively target various cellular substrates. So that will be the way these three talks are linked together. So as I said, I'm going to talk about a very rare human disorder called ataxia-telangiectasia. This is a tragic disorder where the children are born absolutely normal. And you can't tell them from any other child until they're about between the ages of 1 and 6, when they develop a progressive neurologic abnormality that first starts out as clumsiness, and by the time these children are 10 years of age, they're in wheelchairs. So it's a progressive neurodegeneration. But it's actually quite a complex clinical disorder. In addition to the neurodegeneration, the clumsiness and the cerebellar degeneration, they have variable immunologic disorders. They have a significant cancer predisposition, primarily leukemias and lymphomas. They have a liver disorder that leads to increased alpha-fetoprotein levels. They have evidence of premature aging. They have sterility. They have telangiectasias, which is part of the name, which is this little hyperproliferation of blood vessels that occurs on the sclera of the eyes and on skin surfaces. And they have insulin resistance, which is part of what led to the story that I'm going to tell you about today. Now, the gene that's missing in these individuals, or is mutated in these individuals, called ATM, for ataxia-telangiectasia mutated. It was cloned by Yossi Shilo and his colleagues at Tel Aviv University in 1995. It's a very large protein, 370 kilodaltons, and has a PI3 kinase-like sequence in its 4-carboxy terminus. And there's now a whole family of large protein kinases that fall into this PI3 kinase-like kinase family, all of which are involved in stress responses or in metabolism, like the mTOR protein. And much has been learned about what the ATM protein kinase does in DNA damage signaling over the past 10 years. When the DNA is damaged, particularly when there's breaks in the DNA phosphodiester backbone, the ATM kinase is activated, and then it phosphorylates literally hundreds of substrates downstream that affect progression of the cell cycle and affect repair of the DNA. And many of the substrates are involved in numerous processes, and some are very important in cancer, like the p53 tumor suppressor gene, the most commonly mutated gene in human cancer, and of course the BRCA1 gene, one of the breast cancer susceptibility genes that gets so much attention. So ATM is really a major player in DNA damage signaling. But several years ago, we began to think that its functions in the cell were much broader than that. And in part, it was based on a study that we published several years ago showing that in certain cell types, insulin could actually activate the ATM protein kinase, and observations that AT cells have abnormal responses to insulin and that AT patients can have, when stressed, a form of insulin-resistant diabetes. And there was also epidemiologic data that was being generated at the time that suggested that parents of AT patients, carriers of one mutant allele, actually had increased cardiovascular disease. So based on these kinds of observations, we collaborated with Clay Semenkovich at Washington University to ask whether ATM deficiency could actually contribute to insulin resistance in mouse models of metabolic syndrome. And what we found and published a few years ago is that, in fact, loss of a single allele of this ATM protein kinase, which had always been characterized as a DNA damage signaling protein, something involved in genetic instability, actually played a big role in enhancing every aspect of metabolic syndrome. The central adiposity, the blood pressure increases, the insulin resistance, and the atherosclerosis. So when we observed that, we decided to ask the question, if loss of ATM could make metabolic syndrome worse, would activation of the ATM kinase improve the disorder? And fortunately, as part of our basic studies on how the ATM protein kinase gets activated in cells, we had made the discovery that chloroquine, an anti-malarial drug that is one of the most widely used drugs in the world, could actually activate the ATM protein kinase at low doses even without damaging DNA. And so in these various models of metabolic syndrome in the mouse, we were able to show that chloroquine treatment, in fact, could reverse insulin resistance. So if you give a bolus of glucose and blood sugar goes up, in the presence of chloroquine, it goes up less. This did not occur in AT-null cells, so in fact, this benefit of reversing insulin resistance was ATM dependent. It also lowered blood pressure, both systolic and diastolic blood pressure. It reduced atherosclerosis in these mouse models. And so we went on to conclude that even loss of a single allele of ATM, so ATM haploinsufficiency, augmented metabolic syndrome in mice. It suggested that even in the human population, loss of a single allele of ATM, which is relatively common in the human population, may be a contributor to the development of metabolic syndrome. And most importantly, the chloroquine, through activation of ATM, could reverse many of the abnormalities seen in metabolic syndrome. And importantly, this also taught us that it's treating it as a syndrome rather than using Lipitor to lower your lipids and using metformin to lower your insulin resistance. Here is a single drug that could work through all these different downstream events. So since chloroquine is an approved drug, Dr. Semenkovich has gone ahead to take this drug into clinical trials at Washington University, and the results of these trials have been quite striking, that in patients with metabolic syndrome, low-dose daily chloroquine improves insulin resistance, improves lipid profiles, and appears to slow down or even reverse atherosclerosis progression. So now he's going on to develop larger clinical trials, potentially combining it with Lipitor, and in my laboratory we're looking for additional derivatives of chloroquine as additional agents that could activate ATM without inducing DNA damage, and that could be used for this most common of human diseases. It affects about 10 to 20% of the population in the US. So while that work was going on, in my laboratory we were also asking a slightly different question, which was one of the frustrating things about the ATM-null mouse, the mouse model of the human disease, is that though the mice are radiosensitive and develop tumors just like humans do, they didn't have this neurodegeneration, and that was very frustrating to to the whole field. So we wondered whether the process of autophagy, which is a major growing area in the field of cellular metabolism, could actually be neuroprotective in this setting. And we wondered if we downregulated autophagy in AT-null mice, whether we could uncover a neurodegeneration phenotype. And so we crossed ATM-null mice with mice that were lacking a single allele of the Beclin-1 gene, a gene that's been characterized by Beth Levine at UT Southwestern and very popular in the field as a focus of study in regulating autophagy. And even though I thought it was a great idea that crossing ATM-null mice with the Beclin heads would lead to neurodegeneration, it turned out to be absolutely incorrect. That rather than seeing mice with neurodegeneration, we saw the opposite. Not only did they not have neurodegeneration, they actually were healthier than ATM-null mice. And in fact, they lived a lot longer. There was a significant delay in the development of their tumors. And so we began to explore why loss of the Beclin allele could lead to decreased tumor development in these AT-null mice. So the first thing we asked, since ATM plays a significant role in DNA damage responses, was asked whether losing Beclin somehow restored normal DNA damage responses, and the answer was no. Radiosensitivity was still seen, that if you would do total body radiation of normal mice at 8 gray, in this particular strain, they survive, AT-null mice die, and losing allele of Beclin does not reverse that. If you irradiate thymocytes from mice, you get activation of a DNA damage response pathway. AT-null cells don't do that. Losing an allele of Beclin doesn't recover that. So why are these mice doing better when an allele of Beclin is lost? As I mentioned, we already had significant suggestions that ATM did things other than play a role in DNA damage responses, such as this role in insulin signaling and in atherosclerosis, and the fact that chloroquine could activate it. So we had a lot of suggestions that ATM played roles outside of its DNA damage signaling pathways. So we began to look at thymocytes from these mice, which is where they developed their tumors, and sure enough found that the mitochondria were abnormal and played a role in these signaling pathways. So if we looked at generation of reactive oxygen species in the mitochondria from these thymocytes, it was significantly elevated, and the loss of a single allele of Beclin partially rescued that. If we looked at mitochondrial content, it was actually significantly elevated in the thymocytes from these AT-null mice, and again, loss of a single allele of Beclin rescued that. If we looked at mitochondrial respiration, the oxygen capacity, in normal mice, there's a set amount, but that in AT-null mice, you have significant increased oxygen consumption, again, rescued by loss of a single allele of Beclin. And in fact, when we looked even further, in further detail at the electron transport chain, we saw that AT-null mice had dysfunctional mitochondria. Even though there was more of them, they were dysfunctional. There's a decrease in the electron transport chain activity, again rescued by Beclin heterozygosity. So one question that came up, is this due to a direct effect of ATM loss, or is it some downstream effect of ATM being dysfunctional in the nucleus? But in fact, we're able to show that ATM could be found in the mitochondria of cells, so it is localized there. And importantly, mitochondrial dysfunction rapidly activated the ATM kinase. So when we irradiate cells, you see activation of ATM and activation of all the downstream targets that I mentioned in one of my earlier slides. But when you add an uncoupler of the electron transport chain, you get activation of ATM, but no activation of downstream DNA damage signaling. So this says that mitochondrial dysfunction by itself can activate this kinase that we also find located in the mitochondria. So what this has led to is the model that loss of ATM now leads to mitochondrial dysfunction, that leads to increased mitochondrial reactive oxygen species, that the increased reactive oxygen species results in an increase in autophagy. The mitochondrial dysfunction, in the face of decreased mitophagy, which is another thing we've observed in the AT-null mice, leads to at least an initial increase in mitochondrial DNA content, but they're dysfunctional mitochondria. In addition, loss of ATM leads to these abnormal DNA damage responses, and it's a combination of the increased reactive oxygen species, the mitochondrial dysfunction, and the altered DNA damage responses that is contributing to the development of cancer. So it's much more complex than we had ever suspected with ATM loss. And the loss of an allele of Beclin rescues the mitochondrial dysfunction, but does not impact on the DNA damage responses, but significantly reduces cancer development. So what that tells us is that the mitochondrial dysfunction is a significant contributor to the cancer development in these patients and in these mice. So the mitochondrial reactive oxygen species abnormalities are an important contributor to cancer development in these mice, perhaps being additive with the DNA damage response abnormalities. That Beclin-1 has a role in modulating mitochondrial function that we think is separate from its role in autophagy because it's a direct rescue. And we draw the conclusion that ataxia-telangiectasia is much more than a genetic instability disease, but actually should be considered a mitochondrial disease. And so if one thinks about this energy source in the cell, the mitochondria, we know that it's involved in many different metabolic processes, and that when the mitochondria dysfunction, it can contribute to cancer, it can contribute to diabetes, it can contribute to aging, and contributes to neurodegeneration. And strikingly, as I mentioned when I started, all of these phenotypes are seen in AT. So I think this is just a wonderful example of how studying a rare human disorder and linking it now to function of the mitochondria, the energy-producing organelle in the cell, contributes to many human disease phenotypes.